Welcome everybody to WeScience. At WeScience, our goal is to promote local science on a global scale. Today we have a special guest, Dr. Vincent Kuek. He's a scientist at the Kids Research Institute, Australia, here in the city of Perth. Thank you, Vincent, for accepting my invitation to be part of this podcast and for sharing your journey as well as your science and your vision for your research.
Thanks, Carlos, for having me.
Thank you, Vincent. I just wanted to know what triggered you to become a scientist. What was your main inspiration?
Well, my inspiration arises from my own experience when I was growing up as a child. When I was 6 years old, my brother was diagnosed with Hodgkin's lymphoma at 9 years old, and at the time, there was no available treatment for this type of lymphoma in my hometown. So my parents brought him to Singapore for the treatment, and after the treatment, he was in remission for about 5 years, and the oncologist actually said that he was actually cured. So then we migrated to Perth in Australia, and then he had another relapse as an adult of the same disease. So then he had another round of chemotherapy here in Perth, and he's now in remission for many years now, which is good for him. But that experience really inspired me, having seen my parents gone through the financial and emotional struggles that made me want to go into this path to become a medical researcher, and specifically into childhood cancer research to find a better treatment for these children.
Yeah, and in particular for scientists, we become scientists for different reasons. Some of us, is just about making an impact in people's lives. Others, they just wanted to discover new things. But particularly, it's very rare to find a scientist who has a personal story linked to cancer, particularly to the diagnosis, because it's terrible for patients and for families, particularly when it's young kids. It can be devastating. At the time, there was less treatments, but now there are better treatments, and in some cancers they can be treated, and in some of them they can be cured. But we still have a lot ahead to discover. So, and it seems for you it's like a full circle moment. Is that you experience being like a relative of a patient, and then now you're a scientist. You are on the other side making discoveries.
Yes. And in a way, I feel that this is the job that I have now as a scientist is very rewarding. Just doing something, just trying to make a difference, really, for these children with cancer.
So, Vincent, with your motivation, so you started your bachelor and you started working on cancer. Could you please tell us a little bit more about that?
Sure. So I did my Bachelor of Science in Perth at the University of Western Australia. So I did biomedical science degree for 3 years, and then I did 1 year of honors. And then after that, I went on to do PhD at UWA in the field of bone research. And that's when I learned a lot about the importance of the microenvironment in the bone and different cell types that can control the healthy bone balance. And also when these cells, when the machinery goes wrong, they can lead to different bone diseases. So that was a very rewarding 4 years of PhD for me. And then after I finished my PhD, I then continue on as a postdoc there for a further 2 years.
Oh, wow.
Yeah, so I continue to do research, partnering in collaboration with industry to test different types of drugs to treat osteoporosis.
Okay. So you were working particularly with bone-related diseases, not in cancer.
Yes, correct. But I did my honors in melanoma, in skin cancer, yeah.
So then you were working. Could you please share a little bit about your research, your PhD work on bone?
Yeah, sure. So my whole PhD is about finding the types of proteins that can control the bone homeostasis, particularly proteins that can control the bone, the cells that produce the bone, that is called osteoblasts, and also another type of cells that can destroy the bone, called the osteoclasts. So my PhD project was about looking at and see the role of different types of proteins that can control the balance between the osteoblast and osteoclast. Because in the bone, if you think about it, the bone is in a balanced state in adult
Mm-hmm.
at least. And when you have too much osteoblast making bone and not very little osteoclast, you get osteopetrosis, which is too much bone. But if you
Mm-hmm.
have a scenario where you have more osteoclast and less osteoblast, you get osteoporosis. So my PhD research project was about finding the proteins that can regulate this balance. And then the other part of my PhD is looking at the proteins that can control blood vessel formation in the bone, a term called angiogenesis. So because blood vessel formation is important to supply nutrients and
Mm-hmm.
oxygen to the microenvironment of the bone to have a healthy microenvironment. So that was also part of my PhD. Yeah.
When you mention microenvironment, I know that there are different cells types being there. Could you please describe what is a microenvironment?
Yes. So microenvironment is basically like a neighborhood. So if you think about your house, you have many neighbors living around you, and they are all very harmony, to you. And you're living in a harmonious state. And then if something goes wrong,
Mm-hmm.
your one of the neighbor might become angry at you, and then you might have conflict with them, and this
Mm-hmm.
is when you get diseases. So this is the same thing exactly in a microenvironment where all the different types of cells they are living next to each other, and they're talking to each other as well. But then when something happens, like for example, if one of the cell types have some kind of genetic mutation, then you might get bone diseases or indeed cancers. Yeah.
That's great.
Yeah.
Yeah. So you were mentioning about this word homeostasis. For some people, it might be very very hard to understand what homeostasis is. It is a very hard word to pronounce. But could you please describe? For me, what I understand about homeostasis is an equilibrium that you
Correct.
need,
Correct. Yeah.
as you were saying, in terms of the neighborhoods.
Yeah.
You want them to be all happy. Is that one that is the status where everyone is
Correct.
balanced? There is no many. There is cells like eating the bone or the other cells creating the bone. It has to be a balance between them,
Correct.
and that's where the equilibrium occurs. So you said, so you finished your PhD there, and then you said that you did some postdocs. You did a postdoc there in the lab
Hmm.
in University of Western Australia. So what made you move from the PhD to the postdoc?
My passion really for medical research is what's driving me. That was also why I did my PhD in the first place. Is that I'm always curious as a scientist, and I always
Mm-hmm.
want to know the answer to questions that people that are either not interested in finding out, or they are uh, not qualified to find out. So for me as a scientist, I know the skills, I have the techniques, and I can pursue this and find out the answers for them. So that's, yeah.
How do you study bone diseases? I imagine that you have preclinical models where you can test different components and trying to understand what is the variation between the different cell types.
Yeah, well, there are actually two different ways to study bones.
Mhm.
One is what we call in vitro, which means that you take cells out from the bone and put it into the lab in a test tube, and then you grow them up, and then you do different things on them. Like for example, you can test different drugs on them and see whether or not these drugs can affect the osteoblast or the osteoclast. And also you can look at the gene changes as well in these
Mhm.
cells. And then another way is what we call in vivo. So that's where we can use preclinical models, and then we can use different types of imaging techniques to actually visualize the cell in these models.
Okay, Vincent, you were very happy here doing your postdoc after your PhD. What made you change your field?
So I was working for about 2 years as a postdoc with my PhD supervisor, but At the end of the 2 years, my contract was finished because there was funding issues. So I had
Hmm.
to basically go and look for other opportunities.
Yeah. And then how was the gap between you finishing with the first postdoc and then jumping to the second postdoc?
Oh, fortunately, it wasn't too long before I found my next job, which is the current job that I'm in.
Hmm.
So at the time, it was in 2019 December when I finished my contract, and then um, before I finished, I was actually working on another project that is that has the collaborators from the kids, which is my current lab.
Hmm.
So then that is the connection for me to get my next job. So I approached the supervisor of the lab, which is my current supervisor, about a job position. So then they created one for me because they also at the time they received new funding in their
Hmm.
lab. So I was very very lucky in that sense that it wasn't too long for me being jobless. So I jumped straight in after 2 years after I finished my previous contract.
Okay, Vincent. So you transition from your first postdoc to the second postdoc in the Leukemia Translational Research Group at the
Mhm.
Kids. How was the transition for you, coming from the bone biology group, then jumping into leukemia?
Well, in a sense, it wasn't too difficult, I guess, because in the leukemia research, I'm still working on the bone
Hmm.
because leukemia starts developing in the bone.
Hmm.
So, in a sense that the work that I am doing now is still very much using the same techniques and all that
Hmm.
as what I did previously. So, it wasn't very difficult. I guess the only challenge was that when I joined the new lab, that was also the start of COVID. So
Hmm.
the whole institute was locked down, and then I was sent home for about 3 months. So I had to just write review during that 3 months rather than doing any lab work. But it was fun writing. I wouldn't
Okay.
complain.
Yeah. So you mentioned before that you were working in bone microenvironment, and
Yes.
then it was a natural transition for you to
Yeah.
becoming into the leukemia.
Yeah.
Could you please tell us a bit about leukemia? Why leukemia? A lot of people, when you go into dinners and people start to asking you about leukemia. So could you please expand about leukemia and why it's linked to the bone?
Yes. So leukemia is basically a blood related cancer. So it develops when your blood cells become rogue, if you know what I mean. And when something wrong happens in these cells, they will start growing and dividing uncontrollably. They will eventually overtaken the whole bone marrow and then also spread to other sites of your body, like for example, lymph node and spleen in the blood. So yeah, that's leukemia starts from the bone. That's why the skills that I have now it's very very relevant to studying this disease.
Vincent, you transition from your PhD group in working in bone disease and biology to work on leukemia, particularly in the Leukemia Translational Research Group at the KITS. Could you please expand, and particularly you're working on childhood leukemia, could you please expand on why it's important to research and study childhood leukemia?
Yeah, sure. So in children with leukemia, although their survival rate is a lot better now compared to say 50 years ago, but they are still suffering from debilitating adverse effects or side effects associated with the chemotherapy treatment, and they often carry these side effects lifelong. So, for example, after the children finish the chemotherapy. Chemotherapy is very toxic, as you know. So
Mhm.
then it will cause a lot of different effects. For example, fragile bones or osteoporosis. That's one example. Another example could be that they are also aging a lot faster as well compared to uh, normal children without leukemia.
Mhm.
And these children, they are also often have a lot of emotional struggle or trauma associated with the treatment itself. And there's just a lot of it basically will change the these children forever. So our goal is here is to find a way to prevent the development of this adverse effect, or at least minimize the impact of it, so that these children can live a more normal life as they are growing up.
Yeah,
Yeah.
and I also leukemia scientist, and
Mhm.
leukemia is a very terrible disease, particularly because it's the most common cancer in children, and also it's also the second deadliest. So there is still a lot of work on discovering not just effective treatments, but also safer treatments where the
Correct.
kids, they don't suffer all those different toxic effects that the chemotherapy or the treatment does. Of course, there is a new therapies that are coming along, immunotherapy as well,
Mhm.
that are bringing a better hope to the parents as well as for the kids.
Yes.
So it's important also to investigate the not just the origin of the leukemia, but also ways that we can treat the leukemia in a safer way. I think despite there is a 90% of survival, there is still those ones that survive uh, still alive, but there is still suffering some consequences from the treatment. It could be mild, it could be high. So our goal as a scientist is, and I know that you share the same vision and mission with me, along army of scientists that are working on leukemia in kids as well as in adults, is be able to improve the survival rates of those ones that are in a higher risk of dying from the disease. So I just saying this, Vincent, because before there was mentioned to me about the survival rates are 90% in kids. That's why we cannot provide you funding because it's 90%, and then we just see numbers. Could you please share your opinion on that statement?
Well, I actually agree that the survival rate now of 90% is very good compared to
Hmm.
say 50 years ago. However, there are still that 10% of children that are not doing very well. And these are the children that we have to focus on really, because they are also the ones that suffer the most side effects. So, as an example, within these 10%, most of them are infants, because infants' leukemia is the hardest to treat out of all leukemias.
Hmm.
And they often get diagnosed when they were few months old, and they had to go through all these intensive chemotherapy treatment that are really really having a bad effect on the development at
Hmm.
that young age.
Hmm.
So these infants, as they grow up to become adults, they will carry all these side effects pretty much for the rest of their life. So and another common effects from the treatment is also secondary cancer as well. So because of the chemotherapy, have an increased risk of developing secondary cancers growing up as an adult. So this is why we have to, despite the great survival in the 90%, but these 10% they are not still doing very well, and we have to focus on
Hmm.
finding better treatments for them.
And sometimes people just associate it with numbers, and they're seeing
Yep.
numbers 10%, 90%, etc. But we had to go deeper into that because we are talking about lives of children who are in a developing age that can, because of the treatment and different things, can have different, like a very severe consequences to that. And you mentioned about leukemia is very hard to treat. One is because of the different side effects, but also because we are talking normally the treatments are more on adults. They are standardized more on adults because there is a higher number of patients being diagnosed with cancer that are adults, and that's why be able to develop regimes or treatments to be able to bring that treatment power, if we wanted to say in a way,
Mm.
from adult to a child is very tricky. Oncologists and the clinicians are the ones that are making the decision, and that's why it's important to sort of understand, that it's not, we cannot just take the full dose of a adult and bring it to the children, because otherwise they will also be put in a risk for doing so. So you mentioned, Vincent, you're working on bone microenvironment in leukemia. Why? What is the rationale for you and your team to emphasize on the microenvironment?
Because firstly, leukemia develops in the bone marrow. And
Mhm,
secondly, these leukemias, they can interact with the neighboring cells,
mhm,
and they can change these cells' behavior so that these cells become more friendly to the leukemia, and they can provide different types of supports, like for example factors that can make different types of proteins or factors that can cause this leukemia to survive better and grow better.
mhm.
So this is why it is important to look at the bone marrow microenvironment as a whole neighborhood, and we have to find therapies that can target not just the leukemia cells themselves, but also different types of cells that are communicating with these leukemia cells, so that we can uh, not so that we can better treat these children and also their side effects as well.
Okay, Vincent. So you're working on you were just mentioned about bone microenvironment. I just wanted to know what is specifically your research about on changing particularly microenvironment, and why is so pioneering about.
Because in these children with leukemia, they often develop bone conditions, like for example, osteoporosis. And remember when previously I was talking about how in a healthy settings, we have the osteoblast and the osteoclast in a balancing state, but in the microenvironment where leukemia grows, there is this imbalance of more osteoclasts and less osteoblasts that results in bone loss in these children. So these
Mhm,
children, they have increased risk of developing bone fractures either during the treatment and also after the treatment. So they will still have that risk for many years. So that's why it is important for us to not just focus on targeting the leukemia cells themselves, but
mhm.
also the neighboring cells, like for example, osteoclast, how to prevent bone loss that happens in these children. So targeting the neighboring cells of the leukemia is just as important as targeting leukemia themselves.
So if I understand correctly,
Yeah,
so basically what you wanted to do is the leukemia cells are growing exponentially in the bone marrow. So that is causing the bone, all the microenvironment to be in balance.
correct.
Then what you wanted to do is, in addition to the treatment, the conventional treatment that the kids are receiving at the moment, is be able to sort of also add an extra therapy that is able to control the loss of bone caused by the leukemia cells, right?
Correct.
Because there is an imbalance in the leukemia cells are the ones that are destroying the synchrony and the happy environment of the
Correct.
neighborhood. And basically, what you wanted to do is like, yeah, I wanted to treat the leukemias, yes,
Yes.
but also I also wanted to control the bone loss that is induced by the leukemia as well, right?
Correct. Exactly. And our previous research even showed that by targeting the cells
Mhm.
that are communicating with the leukemia cells, that can also improve the actual chemotherapy that targets leukemia cells themselves. So that is a win-win. First, you can treat the side effects, which is the bone loss, and secondly, you can also improve the chemotherapy as well. Because by treating the side effect, you are also indirectly making the chemotherapy better.
That's great. That's really amazing because what you guys doing is not just creating an extra treatment. Of course, we want safer treatments, but you are complementing your main goal is to be able to complement the current treatment so that there is less loss of bone in the kids
Yes.
and they are less prone to fractures.
Yes, correct. That's
So, Vincent, what is your vision about the research that you do? Where do you want to see your research moving towards the future and contribute to the well-being of the patients?
well, firstly, 100% cure rate, because there's so much more we can do for these 10% children that are not doing well. And fortunately, there's a lot of new therapies that are coming up. For example, immunotherapies that are
Mhm.
being trialed at the moment in the clinics for these children, and they are doing quite well. Um, and their survival so far, at least in the early phase of the trial, is looking quite good. So that is a very positive thing for us. But also, my vision is that I want to see these children not having to carry the scars of treatment for the rest of their lives, because these children, they suffer the side effects, and if we can prevent the side effects from happening either during the treatment or after treatment, and they can live a very happy life. And that's what my vision is.
Thank you, Vincent, for sharing the amazing vision that you have. I'm sure that with the hard work that you do, you will be able to fulfill, and I hope 100% of the vision that you have as well, or get closer to that. I just wanted to know in regards a parent with a child with cancer. I just wanted to know from the perspective, probably they are not seeing that the translation is not happening fast enough. That when they see the news about the new discoveries, etc., there is that the research that's happening in the big buildings is still there and doesn't get out from the research institutes. I just wanted to know your opinion on that. What are the main challenges that the scientists like yourself are facing at the moment to be able to bring the vision to fruition?
Well, first of all, in medical research in general, not just in cancer, but in other types of diseases as well, it is a sprint, not a marathon. So, the things that we do right now, it will make a difference, potentially in the next 10 years or 15 years. But because you are doing basic science and basic discovery, it's the foundation of all the clinical trials and all the therapies. So it can be hard to translate from a time point of view, but it is important to do them. That's the first challenge. The second challenge is also the funding issues that we are facing now in the scientific community as well, because there's it's very hard to get adequate funding for the research that we do, and so we have to spend a lot of time writing up grants and applying for grants, and then all that time can be better used to be in the lab to actually accelerate the research to get to our end goal. But that's the reality of being a medical researcher at the moment in this country. Indeed, in everywhere in the world, there are challenges associated with funding.
Yeah, and I agree. Particularly the funding, people might have the understanding that what we do is in a way that everything is controlled. And in here we are dealing with a very complex disease. It's not just ABC. It has all the, as you mentioned, Vincent, that the different players there. It's not just one type of cells there. That leukemia cells, they are very different as well. There are very high diversity on them, partially because of the biology of the tumor, how the tumor evolves. It generates different types of leukemia. Those ones that are maybe be able to be sensitive to the treatment, as well as others who are resistant to the treatment. But then there is also the biology in terms of the bone, as you were saying. So then there's also complexity, and there is also technical challenges as well, and also there is also issues in terms of the new technologies that are coming up that they require significant amount of funding to be able to implement it, because it's very expensive. It's very expensive to do science, and that's why, as the saying says, we need a village. We need a compromise from everyone to be able to contribute. We as scientists, I'm sure that Vincent is doing as well, is driving the research. We are putting our creativity, our energy, etc., to drive the research, but also there is a limitations are in the funding, etc., and the availability of the technologies as well to be able to bring that to be that discovery as well.
Correct. Yeah. And also, I think we also have to look more abroad to philanthropical funding as well, rather than government funding, because
Mhm.
philanthropy, they often have a very specific type of research or diseases that they want to treat, that they can fund that particular type of research. That is also something that we as a research community have to start looking into as well.
Yeah. And also the work that the foundations, for example, the Cancer Council Foundation or the different foundations
Yeah.
that are associated with particular diseases, the work that they're doing is fantastic because they are linking the patients to the scientists as well. And they're also raising some funds to be able to do the science that we do. It's just that there is a complexity in in the biology of the tumor, as well as there is a significant cost associated with all the new technologies that are being used to be able to have a discovery wise has been hidden for many years.
Yes.
So we can make a real treatments that are again very effective, but also safer to patients.
Yeah.
So I just wanted to thank you, Vincent, for sharing that. I just wanted to finalize with this question. So, which factors, and you have been for a couple of years as a scientist, which factors do you think that have contributed to your success as a scientist? I know that scientist is not like a normally like a 9 to 5 job, um, but it requires a lot of efforts from our part. So, could you please share a little bit about that?
One word, passion. You really have to have passion to do what you like to do as a medical researcher, particularly for myself. You really have to have that passion to know that every single things that you do, very small things like, for example, culturing cells to doing preclinical treatments using different types of disease models. Some of the experiments can be technically challenging and also it would take a long time to see the results, but you have to be patient. You have to have passion, and also you have to have the patient to achieve it. Oh, and also when you do this all this work, I often think about the reward. You know, the reward. What is the reward? That is, you're making a difference for children with cancer. So
Mhm.
that is what is driving me every day. And I'm sure this is what is driving everyone that is, working in cancer research as well.
Yeah, and also, for example, mentorship is important, team,
Oh yes,
the
yeah, of
family.
course. Yes. Yes.
How's your experience in terms of the, working as a team in a research laboratory?
Well, you need to have a strong and supportive team that
Mhm.
can provide you with all the resources that you need. The team that I'm in at the moment, they are fantastic. I have lab heads that they are very supportive and understanding of the work that I do every day, and that's also the reason that they give me a lot of flexibility to do my
Mhm.
work. Um, and the culture is very important as well. So I'm very grateful that I'm in a lab where the work culture is very positive. And every single day when
Mhm.
I go to work, I look forward to meeting my colleagues, doing work with them. Yeah.
Thank you, Vincent, for sharing that. I would like to ask you, what advice would you give to those ones that are thinking of becoming a scientist, and the ones that are junior scientists?
Oh, just go with what your passion is really. If you are really interested in science and you're curious and you want to find answers to the questions that no one is interested in, but you are really interested in, just go for it. Find a supervisor that have the resources. That's very important. Have the resources and funding to support you throughout your PhD or postdoc, and also a team that is supportive uh, to you as well. So yeah, just do a lot of research before you join a team. That's important. Yeah.
Great. So thank you so much, Vincent, to spend time with me and sharing your thoughts about your journey on your research. I thank you for taking the time for this. I really appreciate it.
Thank you, Carlos, for having me.
Thank you, everyone, for joining to our podcast. I will look forward to see you in the next episode in WeScience. At WeScience, our goal is to promote local science on a global scale. Thank you so much.